Neta.
Thank
Good
you.
evening, professors and students. With you, Seba Ali, an assistant teacher at Al-Baath University and a current PhD student at Nanjing Medical University. I'm very happy to join with you in this nutrition research webinar 2020. And our presentation today is under the title of the effect of thymoquinone on the reproductive system of obesity model through pregnancy and lactation. Here are the contents of our presentation. And first, let's talk about the effects of obesity on women's health. According to a global investigation in 2014, 40% of women were overweight. Women with obesity are more likely to have a complicated reproduction life. The adverse effects of obesity on women's health are well proven and documented. Obesity can cause fertility disorders like poor conception ability, impaired ovulation caused by unregulated folliculogenesis and poor oocyte quality, delay in lactogenesis, and shorter breastfeeding period, a significant reduction in milk yield. Furthermore, maternal high-fat diet during pregnancy and lactation associates with programmed lifelong tendency to adult disorders. So, to understand the mechanism underlies the metabolic disorders caused by obesity, it's a very important step to fight women obesity disorders. High-fat diet-induced obesity leads to a state of hyperglycemia and hyperglycemia, increased inflammation and oxidative stress, leading eventually to defect mitochondrial biogenesis and function, decreased ATP production, which affects the metabolic function of organs like mammary gland and ovary. On the genes levels, obesity affects organs' metabolic function in different pathways. Obesity suppresses MBK31 PGC1 alpha pathway, leading to mitochondrial dysfunction and impaired energy production and contributing to metabolic dysfunction of ovary and mammary gland. In addition, obesity upregulates AKT-mTOR pathway, leading to follicle growth and loss in ovary and increased tissue inflammation status. Moreover, the insulin resistance status and fat prolactin response caused by obesity affect milk production through suppress the STAT5 JAK2 pathway. So food intervention has been a very good way to fight these disorders. Like we said in other presentation, there is the ketone diet and other supplemented. In our study, we studied the Nigella sativa or black seeds. Is a plant that is very common in the traditional medicine in Asia, like China, in Arab countries like Syria. Thymoquinone TQ is the main active compound in this plant. The essential impact on this quinine was to its antioxidant effect. It's widely studied and proved. It's widely studied and proved for its for its benefits and clinical use in the metabolic syndromes like diabetes and obesity. And a lot of these actions is through the activating of serotonin MBK pathway. The main objective of our study was to evaluate TQ on the reproductive system of obese mice, female mice, through pregnancy and lactation. Examination the ability of TQ to improve redox status and reduce inflammation response, and the examination the ability of TQ to ameliorate metabolic syndrome symptoms such as hyperglycemia, etc. Analyzes mammary and ovarian function through morphology examination and investigation the impact of TQ on lactation performance, milk yield, offspring survival, and ovulation. Also clarify the possible mechanism underlies the TQ effects on obesity through study the genes developed in mammary and ovarian tissue. So here is the design of our experiment. We designed our experiment by dividing the mice into 4 groups: control diet, high-fat diet 20%, high-fat diet supplemented with TQ10, 10% BMM, and high-fat diet supplemented with TQ 20% BMM. We started the experiment with 3 weeks old female mice, and after 11 weeks, we induced conception, as you see. And we sacrificed a subgroup of mice on pregnancy day 18 to study the fetus. On lactation day 15, we sacrificed another group of mice to study the mammary gland. And after 28 weeks of starting the diet, we sacrificed the last group of mice to study ovary. So the methods of our experiment was to evaluate weight, milk yield, mammary and ovary morphology, examine plasma metabolic parameters and oxidative stress biomarkers, and run qRT-PCR analysis to verify the MPK SIRT1 PGC1 alpha metabolic pathway. Here are the results of our study. We can see that TQ decreased weight through pregnancy, but there was no difference, as we can see, on the weight on lactation day 15, and that could be explained by the increased energy demand through lactation. But after 28 weeks of starting the diet, TQ20 decreased the weight significantly. TQ normalized fetus crown rump length on pregnancy day 18 and offspring weight later. And we can see that TQ10, but not TQ20, enhanced early lactogenesis and increased milk yield and offspring survival at 2 weeks old significantly. And by study the effects of TQ on reductive status blood and mammary tissue of lactating females on lactation day 15, we can notice that TQ improved total antioxidant capacity and antioxidants in enzymes activity like SOD, TAT, GSH, and the effect of TQ10 on mammary gland antioxidant capacity is more than TQ20 on mammary gland. By evaluating the metabolic parameters, we found that TQ normalized fasting blood sugar, leptin, insulin levels on lactation and in non-reproduction states. In addition, TQ increased prolactin level on lactation day 15. Also, we conducted whole mount examination for mammary gland on pregnancy day. Here, whole mount examination on pregnancy day 18. We found that TQ10 improved mammary epithelial density, and on the similar manner, by examination mammary glands through HE analysis, we found that TQ10 increased alveolar luminous area. Through HA also, we examined the ovary and we found that TQ20 improved ovulation by increased number of later stage follicles after 28 weeks of starting diet. Our results on the genes levels were the next. TQ10 upregulated MPK SIRT1 PGC1 alpha genes expression, indicating improvement in mitochondrial function and biogenesis, but downregulate TFAM and UCB2 mitochondrial biogenesis genes. In addition, TQ10 upregulated K18, a luminal marker, resulted in improved alveolar luminous development. The decreased level of mTOR and the glucose transporter 1 in TQ10 group could refer to decreased glucose uptake by mammary gland, which could reflect on milk composition, and these will need more investigations. TQ20 enhanced the mitochondrial function in ovarian tissues by increasing RNR gene expression for MPK SIRT1 PGC1 alpha and upregulated UCB2 and TFM gene expression. TQ downregulate gene expression for AKT1 mTOR GLUT1 in both ovarian and mammary tissues, which refer to its previously described role as anti-cancer. So as we see, the mechanisms underlies the positive effects of TQ10 on mammary gland and TQ20 on ovary is by activating the MPK SIRT1 PGC1 alpha pathway, leading to improved mitochondrial function, which contribute to relieve oxidative stress and improve metabolic parameters, and eventually improve organ function. TQ decreased TFAM UCB2 in mammary gland and increased both genes in ovary, which could be explained by the tissue specificity of both genes. UCB2 has been linked to obesity, insulin resistance, and decreased ATP production. UCB2 has been proved to play a role in lipid synthesis in mammary gland. From other hand, UCB2 role in the regulation of follicle development and oocyte maturation and equality has been described. And we can see that TQ10 had a better impact on milk yield than TQ20, and that could be explained as next. Milk synthesis is very complex process, undergoes several different pathways, and the balance between mTOR pathway and MBK pathway is very obvious and necessary to launch lactogenesis. This mechanism could explain the beneficial effects of TQ lower dose intervention in establishing early lactogenesis, increasing milk production, and restoring mitochondrial function in the mammary gland of high-fat diet mice by inducing a positive balance between those both metabolic pathway, MBK pathway and mTOR pathway. So as we see, after the diet, TQ10 induced the positive effects on mammary gland, but TQ20 induced the positive effects on ovary. And this study is the first to see this discrimination in the effects of TQ does on different organs. Our conclusion showed that the food intervention via thymoquinone supplemented diet is effective to ameliorate obesity negative impact on reproductive system and improving metabolic function. And TQ induced this positive impact as anti-inflammatory, antioxidant, anti-hyperlipidemia, etc. And the mechanism underlies those effects was activating the MBK SIRT1 PGC1 alpha pathway, resulting in improved mitochondrial and metabolic function in both organs. This study is the first, as I said, that report about this discrimination in TQ dose effect on different organs. TQ showed its previously described actions on ovarian function in a dose-dependent manner, but those actions were combined with a lower dose in mammary gland. The future prospects after this study, this research is a strong clinical proof on the effectiveness of food intervention in treatment of obesity. Like other professors explained and said, it could be like supplemented food or a different diet like ketone diet or something else, but it's the food intervention is proved to be very effective in controlling and lose weight. Also, and targeting mitochondria through MBK SIRT1 PGC1 alpha pathway, a very therapeutic strategy that could be studied in this supplemented food or different diet. Herein we provide more evidence about the TQ effectiveness in the field of women health and fertility. Actually, there is a lot of study about the positive effect of TQ on the fertility of women and reproductive disorders. So this is additional study that prove the effect of TQ on fertility, and it's the first study actually to study the effect of TQ on lactation and milk yield. Food interventions with TQ supplements could be promising in the future of dairy cattle industry, as we see a lot of industry are seeking for something to increase the milk yield in the milk industry, and this could be another possibility. More investigation need to be done about the effect of TQ on fetus growth, milk composition, and other possible mechanisms that explain its role. Thank you very much for listening, and I'm ready to, if you have any questions. And thank you everyone for listening.